We often speak of trauma as a psychological weight. Biologically, it is a physical failure of internal structure. Major trauma functions as an acute physical event. High-intensity stress signals impact the brain at a cellular level, causing measurable damage to the delicate tissues. Within our neural architecture, trauma can physically sever pathways used to process information. Robust communication lines suddenly go dark. This breakdown isolates the amygdala. Without its usual connections to regulate it, this alarm system begins firing in a state of constant hyperarousal. The brain enters a highly chaotic state, where the alarm remains locked in the on position. The body stays primed for a threat that has already passed, unable to find the signal to stand down. Because the original neural pathways have been physically altered, the brain cannot simply revert to its previous state. It must engineer a new way to function. To prevent total system exhaustion, the brain requires a biological intervention to break the cycle of the perpetual alarm. We possess an inherent biological machinery specifically designed to regain control when these internal systems begin to fail. The prefrontal cortex, the brain's center for logic and regulation, is tasked with executing this recovery plan. The prefrontal cortex initiates an overwrite sequence, sending signals down to suppress the amygdala's panic loop and restore order to the system's electrical frequency. This top-down regulation does more than just quiet the psychological distress. It acts as the mandatory chemical trigger for the physical repair of the brain's cells. Re-establishing emotional regulation provides the necessary biological environment to start reconstructing the severed neural architecture. At the cellular level, the focus shifts from managing the alarm to the actual physical reconstruction of the brain. When the prefrontal cortex intervenes, it stimulates brain-derived neurotrophic factor. This protein floods the damaged areas, repairing the cells. The particles bind to broken neurons, allowing connections to fuse and solidify. New pathways branch out, routing information around the original damage. This machinery operates continuously, reinforcing these new roads to handle the cognitive traffic. The brain does not simply patch over the damage. It uses the site of the trauma as the foundation for building these reinforced alternative pathways. Once the brain has rebuilt its architecture, the new system is structurally distinct from the one that existed before the trauma. This architectural shift reflects a brain that has physically adapted to its history. These BDNF-reinforced pathways can increase the brain's future capacity to process stressors, utilizing the new pathways to maintain regulation more efficiently. The cellular damage caused by trauma creates the exact gaps where these new, more resilient connections are eventually anchored. By engaging the brain's regulatory machinery and neuroplasticity, we have the biological capacity to rebuild ourselves after trauma, emerging with a recalibrated internal structure.